Okay, so we're doing a gel electrophoresis lab, and so this one is the worksheet that I gave you guys yesterday for those of you in my class on Schoology, and this is on colon cancer. This isn't your typical gel electrophoresis lab where we find out, like, figure out who's your baby's daddy or that kind of thing. In this lab, we're actually looking for what? Somebody know? Yeah. So the gene that we're talking about is the HNPCC gene. It only happens in, I think, roughly 3% of colon cancers. And this is a genetic gene. So, you know, a lot of cancers have a genetic component or an environmental component, and this one is primarily genetic, okay? 3% of people that have colon cancer, about 160,000 each year are attributed to this. So there's a strong hereditary component. If you skip down on that third paragraph right here, it says how is it inherited? Does anybody remember?
It's inherited genetically.
Yeah, it says it's inherited genetically. That doesn't tell me anything. Third paragraph.
One has to come from mama and dad.
You don't have to get one from mom and dad. It's actually a autosomal dominant trait here, is what they say. So men and women are equally likely to be affected, so it is not sex-linked. It is not just, you know, if it's sex-linked, it's usually on the X chromosome, and it most of the time affects men. Things like color blindness, or for those of y'all who watch Lorenzo's Oil, ALD. There's a number of different sex-linked disorders. This is not one of those, so it's on a different chromosome. And then, so what is gel electrophoresis? Who knows?
It's kind of like a way of classifying DNA, like comparing it, sort of.
Yeah, so we're comparing DNA. So what we're using today, these are called flash gels, and the DNA is in here. Let's see if I can slide one up. So, well, the DNA is not in here yet. This is actually the gel. Some of you in class may have used a gel that's like a thick jello, like an agar solution, but it works basically the same. You can see in the end, these are the wells, so this is where the DNA is gonna go. And basically you're gonna take your pipette, we've got some DNA in boxes around the room, and I'll try to take you around to one of the setups and show you. But you're gonna take your DNA and you're gonna put it in the wells. And so, how do you know which well to put the DNA in? Good. Flip over to the back side of your worksheet. On the back side of your worksheet, there is 1 through 13 listed on here. And so there are 13 wells. So these are, one is the ladder fragments, which is going to be like, for a comparison to know how big the DNA is. And then the second one is a control that we're going to use to compare. The control is going to be like the individual without the gene. So that's going to be the normal one. And then we're going to compare the other individuals in this family to see if they have this gene that causes this type of cancer. Okay? So the DNA are in little boxes, the little white boxes you see around the room, and we're going to split up in a minute, and then I'll get you to the stations and we'll get started. But I kind of want you to intro to know what you're doing. And then also, these are what the flash gels go in. Basically, once you're done loading the DNA, you will take it, you insert it in here, and you'll turn it on, turn this on. I'll tell you how to do that in a second. And there's a light right here, and so you can see the DNA that, as it runs through here. And that's pretty much all there is to it. Now, from yesterday, remember though, we've got the DNA over here by the black probe on this end. Actually, they're both black, aren't they? Well, on this end, what charge are we going to have hooked up to this? Negative. So you want your DNA running from, the electricity running from negative to positive, because DNA is negative. Two negatives do what? Repel one another. And so basically you're kind of pushing the DNA through here as this negative electric charge goes through your gel. And you'll see the DNA run through here and spread out. And once we're done, I'll show you all some pictures of what the DNA looks like. It's pretty cool. It's pretty easy to read. This is the first time that I've used these flash gels. Usually I do kind of the old school gels where you put the DNA in them. These are easier and they're much, much quicker. So should work out well for us and be able to get it done in this fairly short class period. Okay, this thing right here is what?
Pipette.
This is a pipette. It's a micro pipetter. You can see it has listed how much is on there. And so we've got 8 microliters. And so what you're going to do is this pipette, there are 2 stops. You'll see how my finger goes down and it kind of stops, and then there's another stop. So keep that in mind. We'll talk about that in a minute. Basically what you're going to do when you get your DNA, you're going to mash it down to the first stop, put it in the DNA, and let it up. Okay? Now, when you go to actually put it inside the gel, you're going to take it, you're going to hover over the gel, over that well, you're going to put it down in there, and you're going to mash down to the second stop, which should eject all of the liquid out, okay, all the DNA out. When you do that, do you want to let up on it? No. What happens if you let up on it too soon? It'll suck it back up in there. So you got to make sure you take it out of the gel before you let back up on it, or it will suck it back up in there. Good. So, and you've got a box of pipettes. So these are your tips. What do you need to do after you add a new tip and you put DNA in the well? Why do you need to get a new one? Yeah, you do not want to cross-contaminate your DNA. So every time you use one of these, once you're done, you'll hit the eject button, which is this one right here, and you should have a Petri dish that says use tips on it, and you will put your pipette tips in there, okay? So any questions so far? Super. All right, so let's go ahead and you guys get to your stations, and you need to break up. We'll have a group of 3. Let's do 3, 4, and 3. I don't care how you split up. Just go ahead and split up and get to the station. You need this sheet of paper that has the lab info. You need a blank sheet of paper to write on and something to write with. And if one of you at that station of 3 or 4 will have their book open to 282, that'll help you out a little bit as well. Okay? Go. Get up. Go to the station. Thank you. For those of you that can't see them, which is all of you, they're acting like they're in constant concrete and cannot move. I think they're having a hard time decide who's going to go where. They don't want to leave anybody out either. All right. So what we want to do right now is we want to go ahead and get started and get the DNA in there and get it running so that we will have plenty of time for analysis toward the end of class and be able to analyze what's going on and hopefully make a pedigree at some point toward the end. So, is everybody to the station? Super. All right, you guys on the 4, y'all split kind of in 2. And then you guys with the 3, y'all are going to kind of, since y'all have 3 in a group, y'all are going to work together. You should see 2 white boxes that have DNA in it, okay? They look like this, okay? Inside them, they have DNA. You need to take those 2 boxes, set the DNA out on the countertop in order. Make sure they're in order numbered properly. All right, for those of y'all that read the last paragraph of this lab, we have a number of individuals on here. Like it says, Bob S. is 45 and is married to Jane, 42, blah, blah, blah. Okay, these are the people that we are testing to see if they have this gene in their DNA that causes this particular type of colon cancer, okay? So, that's what we're going to be testing. So there's 13 different wells. And we've got 9 different individuals we're going to be looking at here, it looks like, I think. Because the first 3 things are ladder, control, patient. Actually, there should be 10 different individuals. Okay? So inside here, we've got our DNA they're using. It's inside these things. Take them out, line them up in order. Y'all got them all? This is the easy part of the lab, people. Let's go. Okay. Now, all of you need to gain a little experience doing this. So, if you want to take a shot at, let's just get a pipette out. Get your pipette. Be careful with these. They cost about $250. They're expensive. Okay. Get your pipette out. Are y'all ready? What are y'all doing? in order. It's 1 through 13. It's not nuclear physics.
Is everyone ready? Yes.
Okay, well, you need to turn around and pay attention. So, the gel is laying there. It's in front of Miranda, okay? So, you need to orient that gel where you're looking at it like this. So, this is the left side over here. Right, let's do it like this. So, people at home know kind of what we're doing here. So, there's a gel. All right, we don't need all this information. So, here's your well number. Here are your different wells, 1 through 13. Okay? You've got negative coming in at this end, and your positive probe is at that, or electrode is at that end. Does that make sense? Okay, so you need to orient it where you're looking like that. Now, here's what you're going to do. You're going to take your pipette. Let's all practice in your group real quick. So you'll take your pipette, practice mashing it down to the first stop and then mashing it down to the second stop. So you can feel that resistance at the first stop, but you can continue mashing it down. Okay? Everybody feel that? Because you all are going to do this. We good? Everybody feel it? You guys are actually in this group of 4. Y'all are going to do the 8 through 13, and then you guys, Tay, y'all do the 1 through 7. Is that okay? Okay? So that way y'all can split it up. The rest of the groups, y'all are just going to take turns. So, on the pipette here, so y'all felt the 2 different stops. So, take a tip, get it on there just nice and firm. Okay? Now, I want you to practice holding over the well and putting the tip down in there. Now, these wells are not the normal gels that we would normally use. You have to be super careful. Basically, you can jab all the way through it. These are like affixed in this plastic. So basically, the easiest way to do this is to put your elbow down on the table, okay? And to balance yourself with one hand, or you can hold your hand like this and hold over, and then you're going to put that pipette down in that first well and practice mashing it all the way down to that second stop and then coming up and out. We're just doing a practice go here, okay? So practice putting that pipette tip down in number 1, mashing it all the way to that second stop, pull it out, and then you can let go. And what do you do when you're done? Eject. Now, go ahead and let your partners do that if you want to before you eject it, since we don't have any actual DNA in there, and we're just doing the same thing in number 1. Number 1's our just ladder, DNA ladder anyway, so it's not that big a deal. Does everybody feel good about that? Probably the biggest trouble most people will have with this is that they don't get it all the way down in there, and they squirt the DNA out, and the DNA goes all over the top of the gel, and then you got problems. So make sure it's down in there. You're not going to push through it. Squeeze it out gently to the second stop. Pull it out of the gel, and then let go. Everybody good on that? You might have trouble with that. Okay. Hey, I'm filming. Act like you know what you're doing. Okay, so once you're done with that, you can eject it. Practice ejecting into the tip box with the little gray release button. Don't shoot that at anyone. It is pointy. You can lose an eye with that. Yes. Don't shoot that at anyone. Now, all right, here we go. Here's what you're going to do. You are going to start with number 1 and just take turns with this, Katie. Start with number 1. You'll get have your partner, so y'all need to have a partner doing this. One person opens up the vial, and the other person is going to get the DNA out. All you're going to do is open up the vial, the person getting the DNA out, mash your pipette down to the first stop, put it down in the DNA, and then let up, and then take it out. Do not put your tip down in there and then mash down, because you're going to push that DNA up and out. You're going to make bubbles everywhere, and you're not going to get enough DNA. So make sure before you put the tip down in there, you've mashed down to the first stop, put it down in there, let up. And then go over to your well, hold steady, balance yourself, put it down in that well number 1, mash it down to the second stop, pull it out, and then let go, get rid of that tip, and then get the next DNA. Okay? So in each time, we'll just take turns. If you guys want to switch it up, y'all just do half and half, but everybody gets to do a few of these wells. Does everybody understand? And you'll see the DNA down in the well. They've got a dye that's used, so it's pretty easy to tell what's going on. Any questions about that? Once you're through, let me know, and I'll come around and show you how to turn the gel chambers on. and then we'll get started. These are the gel chambers we're using today, or the electrodes that go with them. We're running on 190 volts, which is kind of higher than some of the others, but that's why the flash gels work so quickly. And then they're hooked up here through a black and a white probe. They hook into the, or not black and white, black and red. And then all you do is set your time, how long you want it to run, and hit the run button, and that's all there is to it. This morning it took about 11 minutes, but I was cautioned that you need to make sure you check them after 6-7 minutes every couple minutes to make sure they don't run too far. If your DNA runs too far in here, then what's going to happen is the DNA is going to go all the way through out the other side, and you're not going to be able to have good results. So you don't want to run that DNA to run too far. Like I said, after 11 minutes, we had it plenty. It ran enough where we had good results. Okay, so I'll make a video in a minute once they're done with this, and we can look at some of these results that go along with these 13 things that are on the that are in our DNA trays. Okay, all right, you guys take care. Part 2. So, we haven't talked about pedigrees yet. That's okay. This is what a pedigree looks like. These are individuals, and then these purple shaded in people are people that had certain traits. Okay? So, you guys are going to take your blank sheet of paper, and you're going to start with Bob S. So, Bob S is right here, and he's married to Jane, 42. Okay? So, things to remember when you're doing a pedigree. Here's Bob, here's Jane. Boys are always squares. Boys are squares, girls are circles. I always think of it, boys are kind of square and bulky and girls are curvy, but this is on the internet, so that's all I'll say. So you need to remember that when you're looking at pedigrees. The girls are all the curvy ones and the guys are the square ones. So the line connecting them together means what? Yes, that is their life mate. So they are married, and then this line right here indicates that these are their offspring. So this is one generation, this is the next generation. These are their offspring. And then if you had their parents, you could add a line up and go into their individual parents. Does that make sense? So a pedigree, the whole point of it is to do what? It's to map out a family tree and to trace a certain trait or a gene that's being passed through. So we'll do this in a couple weeks. Like I said, we haven't quite got to this, but this is not a difficult thing to do, and I hope to God you did this in 9th grade a little bit. But we're going to do one for the royal family of Europe. I got a really big family tree, and you know they're all inbred and passing on like 6 fingers and a bunch of random other stuff. But I've got one for hemophilia. And so there's so much hemophilia in the European family tree from like the 16, 17, 1800s. It's crazy. Anyway, so we'll do that one later on. We'll do a very large one. Right now, you're doing this one with the individuals that we have their DNA and we're looking at in the flash gels. And so what I want you to do is read through that paragraph. It's the last paragraph before the gels right here. Bob S. is married to blah, 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 and so forth, all the way down through their kids and aunts and uncles and whatever. And so you guys are going to go through there and make a pedigree, okay? So I would start in the middle with Bob and Jane and then work your way out until you have completed that. Does that make sense? So and if you'll just write their name in small letters out beside it, that way underneath, then we can go back and track and see who had the trait and who didn't have the trait for the HNPCC caretaker genes, okay? And so basically what's happening here with this type of colon cancer is that individuals are, it's actually short for human non-polyopsis colorectal cancer. I can't even say it. Y'all probably heard of polyps before. What are those?
Like little cancerous, like
Yeah, it can be cancerous or precancerous little nubs or things that are hanging on inside your colon. It kind of looks like, she drew one up here. I don't know if that's an accurate drawing or not right there, but it's kind of something hanging on and growing inside your colon. And so stuff gets caught in it and cells get multiply. And so a lot of times those develop into cancer. That's what they're looking for. If you guys ever have to have a colonoscopy where they go in with a camera up through the 7th planet, and then they go in there and they're looking for polyps most of the time to see if they need to, if they're precancerous or cancerous, they go in, they chop them out, they look at them to see if there's, you know, any cancer growing in them, and then that gives them an indication. All right, so we've got some gels going off. If you guys will go over there and look at your gels, if you'll hit the button, we should start to see some DNA that's running. But also, when you're through, work on your this is part 14 or whatever. So if you looked in your lab packet here, this is considered an autosomal dominant gene, which means it's not sex-linked, and it's dominant, meaning you should only have to inherit how many of the genes from each parent to get the disorder? 1. When you talk about most disorders, a lot of them are recessive, meaning you have to inherit that trait from your mom and have to inherit it from your dad and get both of those in order for that to show up. And so like cystic fibrosis is like that. You could not have it, your partner could not have it, but y'all could both pass on that trait that's recessive to your offspring, and they could develop cystic fibrosis. This is considered an autosomal dominant. Autosomal means it's not on the sex chromosomes. Now, there's a caveat to that, though. And if you look in that last paragraph on the bottom left of the first page here, it talks about this. Cells that become cancerous have inactivated both copies of the mismatch repair gene, a finding that is characteristic of recessive disorders. So what happens here is, yes, you do only need to inherit one of these for this type of cancer to show up. But in actuality, there's a caretaker gene that can be on the other one that you didn't inherit. And if that's still in play, it will act as a fix. It will help to fix any mutation that's there. And so what happens here is, even though you only inherit one, usually when the people get cancer, you inherit one of these bad genes, and then in the other gene from the other parent you got that's good, one mutation is inherited because the cells of the colon are dividing so rapidly. So remember, this is inside your large intestine. Cells are dividing, have to replace each other all the time, like in your stomach and throughout the digestive tract. There's a high probability that a single colon cell will acquire a mutation in the other copy of the mismatched repair gene. In this way, inheriting a single mutation leads to a very high probability of cancer development. So what it's saying there is, because they multiply so often, probably every day or two, that all that has to happen, one of those cells in one of those good genes that you have, if one of those mutates, then it's not going to act as a caretaker gene and fix any of the other bad genes. It's not going to be able to. So you wind up with a genetic component to this, and then also an environmental component that winds up, a mutation that happens because of that. And so this leads to that high probability of cancer that occurs. So what you guys are going to be doing next week on the Pedigree is we're going to go back, we're going to look at your gels, and we're going to see who actually has these mutations. And I'm going to tell you how to interpret the gels next week. And then we will look at how to map that out in your Pedigree and see who is passing these traits on to whom. Y'all take care.